thinking about blood, we should first start by thinking about the functions of the cardiovascular system or the circulatory system. And the first function is going to be to transport things around the body. And what is it transporting? Blood is transporting anything that's got to get from one place to another. So it's transporting our respiratory gases. It's transporting nutrients and wastes from one place to another. Transporting hormones. Basically anything that's got to go from point A to point E, or point B, or I guess E. We've got to get from one place to the other. How we're going to do it is in the blood. So that's the first function. The next function of the circulatory system is to help with regulation. And what's it going to regulate? It's going to help regulate a couple things. First, it'll help regulate body temperature. Body temperature has to be kept in a pretty tight homeostatic range, not the most tight homeostatic range. You can drop a couple degrees, you can have a fever for a couple degrees, get hot and not have too many problems, but we have to stay kind of around that 98.6 area, because if you go too high or too low, you're going to start changing enzyme activity. If you start changing enzyme activity, then you start degrading proteins, you start changing the rate of chemical reactions, and you have big problems overall. So the circulatory system helps with the regulation of body temperature because your blood can pull heat away from your internal organs and bring it to the surface of your skin. If you're too hot, then we can turn on our blood or our sweat glands and lose heat through evaporative heating. If we're too cold, we can dilate those blood vessels in areas that aren't absolutely necessary, like your fingertips, your skin, and things like that. We can constrict those blood vessels and bring the blood, the warm blood, in to maintain that body temperature if it gets too cold. So our circulatory system is going to help with the regulation of body temperature by either helping us to lose heat when we get too hot or to keep heat when we get too cold. So we'll say blood vessels can dilate or constrict to allow for temperature changes. Another thing that blood is going to help regulate is body pH. pH is another thing that has to be kept in homeostatic range, and it does have to be in a very tight homeostatic range. The pH of blood can range from 7.35 to 7.45. So in general, we're at an average about 7.4, which is slightly alkaline. And we need to keep it in that range for the same reason we've got to keep body temperature tightly controlled, because if you mess with pH, you mess with enzyme activity. You mess with enzyme activity, you start changing chemical reaction rates. So our blood has buffers that protect against fluctuations in pH. And then the last thing that blood is going to help with regulation of is body fluid balance. We'll talk more about the fluid compartments when we get to the urinary system, but basically your blood has to be maintained within a certain level of viscosity or thickness. And if you get too viscous or too thick, the blood we can pull water out of the intracellular fluid compartment, which is not great, so you're more likely to get thirsty and take a drink, but if need be, you can do that. We can pull blood or pull fluid from the intracellular fluid compartment and bring it to the extracellular fluid compartment. So it's going to help keep the osmotic pressure on water to balance between our fluid balance in the tissues and our fluid balance in blood. So we'll say that blood helps maintain osmotic balance or pressure. Those are going to be our 3 regulatory functions, helping to regulate body temperature, helping to regulate body pH, and helping to regulate fluid balance. Then the last function of the circulatory system, or blood in the circulatory system, is going to be to offer protection. And that's because our white blood cells are our immune cells. So these WBCs are white blood cells. And they offer protection against pathogens. And we'll talk more about this when we get to the immune system. And another thing that blood is actually helping with is blood loss. And that's because the proteins and platelets that are needed for clot formation are floating around in blood. So we can help kind of prevent blood loss by forming clots. And we'll probably talk about clotting on Monday. Alright, so we can briefly discuss the characteristics of blood. Blood is red due to this protein kind of complex molecule called hemoglobin. And whether hemoglobin has bound oxygen or not will determine what shade of red it is. So it is bright red when hemoglobin has bound oxygen. So we say the color is due to hemoglobin. We're going to talk about the structure of hemoglobin in a little bit. It'll be bright red with oxygen bound. And then when we've unloaded oxygen in the tissues and picked up some CO2, it'll turn dark red. We'll say dark red after O2 unloading. So it's got a characteristic red color due to hemoglobin. And then whether or not it has oxygen bound is going to tell us whether it's bright red or dark red. The next characteristic of blood is that we have a characteristic amount of blood, or a characteristic blood volume. Males tend to have 5 to 6 liters. Females tend to have 4 to 5 liters. So humans have an average of 5 liters. Also got this characteristic called viscosity. Viscosity is thickness, and blood is 4 to 5 times thicker than water. This is due to the presence of our formed elements and due to proteins that are floating around in the blood plasma. When we look at blood composition, we'll look at the different types of proteins that float around in blood plasma and see which proteins are going to contribute most to viscosity. So we can change viscosity by changing the number of red blood cells or by changing the amount of these proteins called albumins. The next thing about blood is that we have a specific amount of different solutes that we'll find in our different body fluids, and the amount of those solutes in the plasma will determine movement of various solutes from the intracellular compartment to the extracellular compartment. So for plasma concentration, we can say that the concentration of solutes will determine the movement of these solutes into or out of the blood. The next characteristic of blood is that it has a characteristic temperature. It's about 1 degree higher than body temperature. So as it's moving around, it kind of warms places as it goes. The last characteristic we've already talked about a little bit. It's got a characteristic pH, which is the 7.35 to 7.45. That makes blood slightly alkaline, and it has to be kept in this range so that we don't alter enzyme activity. So we'll say 7.35 to 7.45 to maintain normal enzyme activity. When you get too far out of this range, you start denaturing proteins, so you start really messing with the system. Any questions on the functions or characteristics of blood? All right, so to determine blood composition, you can take a sample of whole blood from a person and put it in a tube and spin it down. So they'll put this tube in this thing called a centrifuge and spin it down, and when you spin it down, then the heaviest elements sink to the bottom. And what you find is that about 45% of whole blood is erythrocytes, which are our red blood cells. And they're the heaviest elements, so they drop to the bottom. If we look just above that, we'll have a little layer that's called the buffy coat. The buffy coat is white blood cells. So when you're not sick, it should be about less than, like 1% or less of your whole blood composition. If you're sick, then your buffy coat will be a little bit bigger. You'll have more white blood cells floating around, and that will tell you that you're having some kind of an immune response. Most of your blood is blood plasma. So at the top of this tube, we would find blood plasma, which is this straw-colored fluid. It's about 55% of whole blood. And we'll talk about the specific components of blood plasma in just a minute. So again, our red blood cells will sink to the bottom because they're the heaviest. Next, our white blood cells will be found in that buffy coat. But again, unless you're sick, it shouldn't be very thick. And then on top, we have this straw-colored fluid that is our blood plasma. So what is blood plasma made of? If we look at the composition of blood plasma, it's this straw-colored fluid that's about 90% water. So it's mostly water. And then floating around in that water, we've got proteins, we've got non-protein nitrogen substances or nitrogenous substances, and we've got electrolytes floating around. So as far as our proteins go, we have different types of proteins that we'll find floating around. The first kind are called albumins. These are the most abundant, and these contribute most to blood viscosity. So if we need to change viscosity, if we need to increase blood viscosity, we can make more albumins and put them into our blood plasma. Albumins act as buffers. So these are buffer proteins to help protect against changes in pH. And these, we'll say, contribute most to blood viscosity. When you look outside the formed elements of the proteins that are found in blood plasma, albumins contribute most to blood viscosity. So those are the first types of proteins that we'll find. The next types of proteins that we'll find are called globulins. And there are three types of globulins. We've got alpha globulins and beta globulins. These are going to be like our transport proteins that are transporting around our lipid-soluble hormones and our fat-soluble vitamins. So we'll say our alpha and beta globulins transport lipid-soluble materials. And then the last type of globulin is a gamma globulin. And these are produced by immune cells and are antibodies that are going to help with the immune response. So we'll talk more about gamma globulins when we get to the immune system. The last important protein we need to talk about is one called fibrinogen. Anytime you see O-gen, it's telling you that we're generating something. So fibrinogen will make fibrin, and fibrin is an insoluble protein that helps with blood clotting. So fibrinogen is floating around in your blood, so you get a cut and it can exit the blood and enter the tissue and be converted to this insoluble protein, fibrin, that will help pull the clot together. So we'll say this becomes fibrin to help with clot formation. So those are the main proteins floating around in blood. Of course, if you got a protein hormone floating around in blood, that would be a protein floating around in the plasma. But these are the biggest other ones that we'll find floating around in the plasma. Other things that we'll find floating around in plasma are what are called non-protein nitrogen substances. So those are waste like urea and uric acid. We actually have to have low levels of those materials floating around for homeostasis. Excess levels are removed by the kidneys and excreted in the urine. Those will be floating around in the plasma. Urea, uric acid are waste. We can have amino acids floating around that are just the result of protein breakdown or protein digestion. It's non-protein because they're just the breakdown products of it. So that'd be another thing that we'll find floating around. You can say our amino acids. These are the byproduct of protein digestion, or the breakdown product of proteins might be a better thing to say. So those will also be floating around in the blood. And then the last thing that we'll find floating around in the blood are electrolytes. Floating around in high concentrations in blood plasma, I should say, are electrolytes. The cations that we have floating around are things like sodium, potassium, calcium, magnesium. Magnesium also has two positive charge. The anions that we have floating around are like chloride ion. Who else? Bicarbonate. So electrolytes are ions. They can conduct no charge in aqueous solution. And so these are going to be primarily absorbed from the small intestine into the blood, and we'll find them floating around in the blood plasma. Then in whole blood, all of our formed elements are suspended in the blood plasma as well. So just speaking of blood plasma alone, when we pulled it out, all of those last things we were talking about, but then we also have our formed elements in whole blood. So our formed elements are our red blood cells, which are also called erythrocytes. Our white blood cells, which are called leukocytes. And platelets, which are cell fragments. Platelets are going to be important in blood clotting. All right, any questions about the composition of blood or blood plasma? Okay, and let's talk about our different formed elements, and then we'll talk about blood formation. Erythrocytes are our red blood cells, and they are interesting cells because when they're ejected as mature red blood cells, they're pretty much non-living. They eject their nuclei and they eject their organelles, and it gives them this characteristic biconcave disc shape. Why do you think we would want to eject our nucleus and organelles?
Less energy use.
Right. They don't have, because if you have a nucleus and organelles that are doing a bunch of stuff, then you have to have energy. If you have to get energy, we have to use oxygen. We don't want these guys to use oxygen because they're transporting oxygen. So their big job is to transport oxygen around the body. And so if they're using oxygen, they wouldn't be very good transport cells. So when they're formed in the bone marrow, it takes about 2 days to reach maturity, and in that 2 days they'll be killing off their organelles, and then they eject their nucleus, and then they'll enter the blood and will circulate for about 120 days before they wear out. So their job is to transport oxygen, so they can't really be using a bunch of oxygen. As far as their composition goes, they have a whole bunch of this stuff called hemoglobin. That's what gives them their red color. Each individual red blood cell has about 250 million hemoglobin molecules. And each hemoglobin molecule has they're these big globin proteins. So they've actually got like 4 sections. And then deep within that section, they've got this stuff called heme. Heme has iron in it, so this is why we require iron in our diet. If you're low on iron, you're called what's anemic, and anemia can lead to red blood cell problem or production problems and low oxygen carrying capacity. So the heme molecule is like deep inside that protein, and this is what carries oxygen. So each hemoglobin can carry 4 O2 molecules. So each hemoglobin can carry 4 O2s, and we've got about 250 million hemoglobins per red blood cell. So red blood cells are very good at carrying oxygen. The heme portion carries our oxygen. Each hemoglobin then can carry 4. This globin portion can actually bind CO2. This is not the major transport mechanism for CO2. These guys' big job is to transport oxygen around. CO2 is primarily converted to bicarbonate and transported as acid in the blood. It gets to the lungs, we convert it back to CO2, and we breathe it off. We'll talk more about that when we get to the respiratory system. But we can bind some CO2 to the globin region. So low levels of CO2 can be carried away from tissues on the globin portion of hemoglobin, although the primary job is to carry a lot of oxygen. All right, questions on that so far? So then we'll talk next about blood typing. Blood typing comes from surface antigens that are present on the surface of your red blood cells. So antigens are things that will initiate an immune response. So it's kind of a weird way to use antigens when talking about something that's normally found in your blood, but they will cause an immune response in somebody who doesn't have your blood type. So I'm going to give you some definitions. Antigens in general are things that cause an immune response. So when we get to the immune system, we'll talk about pathogenic antigens, so disease-causing antigens. And then the other thing that we should talk about now are antibodies. Antibodies are proteins that mark something for removal from the system. They mark antigens, so proteins that mark antigens for removal. So these are going to come up again when we get to our immune system, but for now we call these different proteins that we can find on the surface of our blood cells antigens. So we have different types. You've probably heard of the ABO groups. So everybody's got either A antigens on their red blood cells, B antigens, both, or none. So for our cell surface antigen A, it would just be like this protein that we call the A surface antigen, and it marks you as an A blood type. If you've got cell surface antigen A, then your body makes the antibody anti-B. So floating around out there in your blood plasma, we would have anti-B antibodies. Antibodies have two regions that can bind to antigens, and so these proteins will be floating around in your blood, and they're anti-B. So if we look at type B blood, their surface markers are shaped differently. We would just call them surface antigen B. Not a very good one. And if you've got B antigens on the surface of your cells, then your body makes anti-A antibodies. So if you've got surface antigen A present, you make anti-B, which means that if you have B if you get B blood and you have anti-B, or you've got B surface antigens present, you'll start an immune response to that. The antibodies will bind it and mark it for removal from the system. What do I mean removal from the system? When we get to the immune system, we'll talk about the 6 ways that antibodies work to mark something for removal from the system, but basically it's going to, in the case of blood cells, clump them together. And then it's called agglutination, and things that are agglutinated together get pulled out by the liver or the spleen. In this case, it can be really bad. If you get a blood type mismatch, you go in, you're type A, and they give you type B blood, and they give you a liter of it, then you start an immune response, and you agglutinate that whole liter of blood, and all those cells will clump together, and they'll start blocking small blood vessels. So it's very dangerous. It's really important to make sure that you've got a blood type match. If we look at type AB, the blood type AB, people with AB have both surface antigens. So they'll have type B antigens on their surface of their blood cells, and they'll have type A antigens. So what this means is that they do not make an antibody. You can't make an antibody because you have both surface antigens. You can't start an immune response to yourself. That would be counterproductive. Type O blood doesn't have surface antigens on their cells, but because of that, they make both types of antibodies. So they make anti-B, and they make anti-A. Oh, I think I just said that opposite of my colors, but either way they make, anti-B and anti-A antibodies. So when thinking about blood donation, when you're accepting blood, or when you're getting blood, they're not giving you whole blood that has both. They're giving you blood cells. that is free of the plasma proteins that could start an immune response. So, who could take type O? Everybody. Type O is the universal donor. Everyone can take O blood because there aren't surface antigens. So if you make anti-B, we're not going to fight it. You make anti-A, you're not going to fight it. So everybody can take type O. Type O people can only take type O because their blood is making anti-A and anti-B. So they're only able to accept type O blood. So if you're type O, especially type O negative, then Bonfils is calling you every month. Hey, you want to come donate? Hey, we need some more blood. You want to come donate? Yeah, we'll pay you $10. So give you some lunch. So type O blood is very valuable because it's the universal donor. So we could say that this is the universal donor. Type AB blood is the universal recipient. They don't make any antibodies, so they can take anybody's blood. They can take B, well, they can take anybody's blood cells. They can take B, they can take A, they can take O. So type AB is the universal recipient. What types of blood do you think type B people can take? They can take type B or type O. And that's it. They can't take AB because they're making anti-A, and those AB cells have A surface antigens. How about if you're type A, what can you take? Type A or type O. Again, you can't take AB because you're making anti-B, and there are B surface antigens on AB cells. So your lab today is going to have you first predict whether or not things will agglutinate. So that'll help you kind of decide what's going to happen in your lab. Most of the time, people get this exactly opposite of how it should be at first. So just remember, if you are type A blood and I give you anti-A serum, then you would agglutinate. If I give you anti-B serum, you would not agglutinate because you don't have B on you. So the first part of your lab is going to have you guess. It gives you blood types and has you guess whether or not it will agglutinate. And then for the second part, we're actually going to do blood typing. It's not real blood that we use, but it's mocking blood. And then so you'll get an unknown blood type, and you stick different serums on it and determine what type it is based on agglutination. So that'll give you some good practice with our ABO blood types. Any questions on that so far? Then the other thing that we have to talk about is what's called the RH factor, or the RH blood types. So you've probably heard of A positive, O positive, A negative, O negative, and that positive or negative is referring to what's called the RH blood types. RH because it was discovered in rhesus monkeys. Your book calls it cell surface antigen A, but there's actually a group of like 4 surface antigens that you can have that would make you RH positive. So if you are RH positive, then you have this surface antigen D. We'll just say, because that's what your book says. So if you're RH positive, you've got this D group on the or on the surface of your antibodies or of your red blood cells, in addition to your ABO group. But you don't make any antibodies because you can't have an immune response against what you have. So you don't make any anti-D antibodies. If you're Rh negative, then you don't have surface antigen D present. But anti-D isn't something that you make right away. It's not something that you just naturally make. You have to be exposed to Rh factor before you start making it. So if you've never had a blood transfusion and all they have left in the ambulance is O positive blood, then they can give you that without knowing your Rh status. Because the first time it doesn't matter. You're not making anti-Rh. But as soon as you're exposed, you'll start making anti-Rh. So you would not be able to take Rh positive blood again if you're Rh negative. This is kind of like hard to wrap your head around, but if you, except for in this case, I used to have the figure up here, of this condition called erythroblastosis fetalis. So I'm going to come to this blank slide and kind of draw this out for you. So if you've got a woman and and she's like O negative, and she's pregnant with her first fetus that is Rh positive, then the first time she's pregnant it's not a problem. Throughout gestation their blood isn't really joined and there's no real issue. But as soon as she delivers that baby, labor and delivery is a violent and bloody process. So when that happens, she will be exposed to Rh, and she'll start making anti-Rh antibodies. So after the birth of that first child, she'll start making anti-Rh. Then she gets pregnant again with her second Rh positive child. And now she's got those anti-Rh antibodies, so her blood cells will start attacking that fetus. And this can kill the fetus. And so it can be a deadly event that happens. It doesn't have to be. If you know that you're Rh negative and you've had an Rh positive baby, then they can give you a shot of this stuff called Rogam that suppresses the immune response so that that won't happen. But if you don't get Rogam, or you didn't know your baby's blood type, then it can kill that baby. Even with Rogam, it's hard to fully suppress the immune response, so these babies tend to be born with what's called jaundice. And jaundice is when their skin is very yellow. And their skin is yellow because their mom's antibodies have been attacking their blood cells. A byproduct of breakdown of your red blood cells is that heme group gets converted to this stuff called bilirubin. We'll talk about what bilirubin does and where it goes in the normal life cycle of a red blood cell, but if you've got a bunch of bilirubin around, it'll make that baby look yellow. So their skin gets very dark, and you can tell there was a problem. So the Rh factor isn't really hugely important until some situation like this, where you've been exposed to it, you get sensitized to it, and then you start making anti-Rh. If you're thinking about donating an organ to a friend or something who needs one, the Rh factor is less important than the ABO factor. So if you're A positive and your friend needs they're A negative, they can take your A positive kidney and they can kind of hamper this Rh response because it's not something that you're automatically born with. So the ABO is much more important on a moment-to-moment basis, but there are some times when the Rh factor can come into play and cause problems, like in this example is the best example.